Welcome to the Imprint Hub, where we talk about maternal and pediatric pharmacology. Today, we're taking a deep dive into how the body's ability to manage medications changes as we grow, from before birth through childhood, and what that means for drug dosing and drug modeling in pediatric populations. We'll explore how key concepts like ontogeny and allometry impact drug dosing for newborns, infants, and children, and how we use this knowledge to build pharmacokinetic models to predict safe and effective dosing across the age spectrum, especially when real-world clinical data in these groups are limited. To walk us through this, I am joined by Dr. Aislinn Pruss, who brings expertise in both developmental pharmacology and modeling.
Thank you for having me.
Let's start with some pretty basic context with what we mean when we say developmental pharmacology.
So developmental pharmacology is the discussion of how the body is changing as an individual ages. Typically, we talk about the start from birth. Sometimes we talk about before birth. There's something that's called postmenstrual age. Postmenstrual age can be really important for some drugs because the liver develops while a fetus is in the womb, but then after they're born and they become an infant, their livers continue to develop. This is why some premature infants have to be put under a light, because their livers are not appropriately developed in order to get rid of like the bile in their body so they can get jaundiced, and light actually helps get rid of that. And that's because of developmental pharmacology, because their livers are not fully developed yet and they don't have those enzymes to help them break down that bile yet. So they develop that later on. And this is super important when it comes to dosing drugs, particularly in neonatal, infant, and younger children populations, because all of them can be metabolizing drugs differently depending on where they are in that stage of development. And this is called ontogeny. So ontogeny is the development of tissues with time, with age. There's also something else that is important in developmental pharmacology, and that's called allometry. And allometry is basically clearance with size. So it's how the size of something, for example, like how much a person weighs, is going to directly impact how much they break down a drug or how they distribute a drug. So you have to factor in both allometry and ontogeny.
So walk us through the process. In developmental pharmacology, we're often working with very little clinical data from our target population. So how do you take that little amount of data that we have and create a predictive model with it?
You basically ask relentless questions, and then you try and answer all of your questions. And then when you think you're done with your questions, you actually have to ask more. So your first one is going to be, what's my best baseline? So what is the most reliable data that I have in the most stable population in terms of pharmacokinetics and how the bodies work? So typically going to be a standard adult population. So hopefully you have some adult PK data, typically like a single ascending dose or a multiple ascending dose study, typically created by the sponsors of the drug or by the FDA. And that gives you a very standard dose in a very standard population that is healthy, that doesn't have anything influencing any of their organs or any of their pharmacokinetics. So that's like your baseline. And then you build your model around that. So you use like a healthy volunteer population, you input the age, you copy the demographic, you copy the ethnicity, the race, to make sure that none of those are going to confound anything. You make sure that your weights match as well in terms of allometry. And then you also make sure that all of your drug parameters are going to match. And then there's the question of sampling. How good is your sampling? Typically, you want rich data. So you want multiple data points across a period of time. And sometimes that's not always possible. Sometimes you get sparse data sampling where you only get a couple of blood samples and you kind of have to fill in the gaps. And that's where population pharmacokinetic modeling comes into play, and it really helps you extrapolate based on that. But after you deal with that population, then you start to go further into your population. So do I want to know what this drug is doing in infants? Do I want to know what different genotypes in adults are going to do to this drug? What disease states am I dealing with? Is there liver failure? Are my patients on ECMO? What is happening there? And how do I design my model in order to best fit that? And then you have to ask a lot of questions about exactly how is my drug being broken down? What parts of the body is it going into? What is it binding to? Is there a difference in any of those features between men and women, between different races, between different ethnicities, between different ages? So you are just categorizing and stratifying as much as possible, and in order to get the best picture. And then afterwards, you always make a note of every change that you made between all of your populations, between all of your simulations, between all of your anticipated breakdowns and distributions and absorptions of your drug. And you make sure that you do a stepwise process. So if you change all of that at once, you're not going to know what made your model fit better or worse. So you have to go very sequentially, you have to be very patient, and you have to be very diligent in documenting every single change that you made to your model. And then you compare it to your output. And that's the most important thing, is that you have a validation study. So you have that rich data from your patients and you try and replicate it using your model.
So can you talk us through what this looks like in your research?
So we had a patient population, and it ranged from 0 days old to 20 years old. And we had to account for both allometry and ontogeny in our patient population. So the first thing that we did is we just plotted the relationship of clearance along with age and years. And then we looked at clearance versus body weight. And then we incorporated that and we corrected clearance based on body weight versus age. And this gave us a much better idea of what is happening in terms of this clearance. And then next, once we fixed our model allometrically, we were able to move down to ontogeny. So once we figured out how the clearance was interacting with size and then accounting for age, we moved down to a tissue level. And then we looked at how, once you correct for everything else and you only look at age, what's happening with the clearance. And that's where we see this really interesting jump here. So here we've got our newly born infants. And as you can see, there's a wide range here in terms of their metabolism and how quickly they're breaking down the drug that we're studying. You can see that they all start out below 50 liters per hour, and then they spike as they move a little bit past here. So as you can see, they're almost getting up to 150, even over 150 liters per hour here. So you can see this very, very wide range. And then you can see that the clearance levels out, and you can see that there's a lot less heterogeneity later on, which shows that all of the tissues are reaching a rate of similar development. So you've got, in this region, what we could hypothesize is happening is that some of these infants are developing a little faster than others, which could be dependent on how long they were developing in utero for before they were born, so what their postmenstrual age is. And some of it could just be dependent on inter-individual variability. So some, they just develop a little bit faster than others. So that's what I believe that this wider range here is. And it's also indicative of the fact that ontogeny is happening, that these infants are developing. They're starting out small down here, very concentrated amount of liters per hour. So that gives us a good idea of where their baseline is for that enzyme when they're born. And then you can see that enzyme starting to develop because their clearance jumps up and then it slowly normalizes back down to normal. So this is a snapshot of a very wide range of individuals. And part of the reason why there's so much wider of a scale down here in this age group is because the majority of this population was shifted to that age group. There were a lot less patients that were sampled in terms of their pharmacokinetics in this age range. So really from like 10 to 20 years old, that's a much smaller fraction of this population than this population here. So one thing that you might take out of this graph is that everything normalizes back down to kind of a standard rate as you get older. That's not necessarily true. It's just that we have a bigger sample size on this end of the age range. So what we did is we looked a little deeper into that age range, and we expanded this kind of key age group where we saw that big jump in clearance. So between birth and then 2 years old, that's where we really see this jump. Even though there's more heterogeneity, that's where we see the trend line going. So we took line right here, which is, again, allometrically scaled, so we don't have to worry about body size. We're already incorporating that in every single one of these samples. We just need to worry about age and how the tissues are changing with age and how you're breaking drug down better with age in this specific scenario. So we plotted, based on this line, a couple very important key points, and then we incorporated those into a model. So we took that data from our population where we plotted out the clearance, the allometrically scaled clearance against age, to figure out how the clearance was changing with age, the ontogeny. And then we took data points from that and we put it into Simcyp, which is a physiologically based pharmacokinetic modeling software. And that helped us figure out what the fraction for each age group would be of the total metabolism for the enzyme that we're looking at. And so based on that graph that we looked at, we estimated that at birth, neonates have, on average, about 50% of the activity of that normal enzyme. So this is the rate that we calculated based on our equation that their clearance would be increasing by. And then we calculated that the age where they would have really met that max would be like 1.1 years. So based on that, we looked at the fraction of metabolism that was happening with our individuals that we were seeing. Now, one thing that I want to discuss here is that our specific populations and our specific samples that we saw in all patients that were under 2 years old, this drug that we were studying, which hopefully we'll publish about it very soon, we were looking at patients that it's typically used in very sick patients that are undergoing surgery or that are in the ICU. And the only data that we could find that was useful for our model in patients under 2 years was patients that had to undergo something called cardiopulmonary bypass. That caused a big difference in clearance.
So we'll talk a lot more about how extracorporeal life support impacts pharmacology in our next video. But for now, sticking with ontogeny and modeling in these young patients, what kind of sampling and clinical data do we need to validate those predictions?
Even though we were able to very accurately predict in this specific population between hepatic ontogeny, so the ontogeny of the liver enzymes, as well as the ontogeny of the medical equipment that was used to help these infants breathe and help circulate blood through their bodies, we don't know exactly. We have a good idea, but we don't know for sure how to differentiate them because all of our models depend on being able to validate in populations that are representative of our models. So if we separate those two, we don't have a population yet where we could look at that. So that's where you need really good sampling, and that's where ontogenic studies are really, really important. And it's kind of a newer thing that's being studied. A lot of people didn't really look at that sort of development before as much in the past, but now that there are more grants available for neonatal research and infant research, as well as maternal and pediatric research, there are more opportunities for us to figure out really what's going on in the bodies of these little and very fragile patients so that we can figure out step by step as they age what the best target doses are for them, what the best therapies are for them, and how we can really tailor our medications to meet them rather than giving them a one-size-fits-all type of treatment that may or may not work the best for them. And that's my goal.
Thank you so much for joining us, Dr. Pruss, and thanks to you, our audience, for being here. The rest of our conversation with Dr. Pruss about pharmacology and extracorporeal life support, using the most of your clinical data, genotyping, and more should be out soon, so we look forward to seeing you there. Let us know in the comments below if you have any topics you'd like for us to cover, and we'll see you next time.